I am Vivek Pattani. Hi everyone. Today we are going to discuss immunity and immune system with Mr. Sagar Sonara. Please.
Myself Sagar Sonara, and hereby we are coming with a conceptual learning about immunity and immune system. So here we go.
Okay. Thank you, Sagar. And points which we are going to discuss today are basically types of immunity, such as natural, acquired, herd, innate, as well as the specific immunity, structures and functions of the immune cells, stem cells, T cell, B cell, NK cell, macrophages, neutrophils, eosinophils, basophils, mast cell, and dendritic cells.
And then we also discuss on the structures, functions, and the properties of the immune organs like bone marrow, thymus, lymphoid, spleen, GALT, MALT, and CALT. And at the last, we will also discuss on the characters of immune system.
Okay, thank you, Sagar sir. First of all, what is immunity? Immunity simply can be defined as the way in which the body can protect itself from the invasion by pathogenic microorganisms and provide defense against their harmful effect. Simply, a physiological function of the immune system is to protect against the infectious microbes and the non-infectious macromolecules. There are basically two types of immunity: innate and acquired. Innate immunity is also known as non-specific one, and adaptive or acquired immunity is also known as specific immunity. This can be further divided into cellular and humoral responses. Both could be either natural or artificial. So moving forward, innate immunity. So which are the barriers or which are the points which are covered in the innate immunity? Okay, those are anatomical barriers. Those are basically anatomical barriers. Okay, uh, phagocytic barriers, blood proteins, cytokines, etc. So basically, what are anatomical barriers? Okay, such as skin, okay, mechanical barriers, mucous membranes, phagocytic barriers such as neutrophils, macrophages, natural killer cells, etc. Okay, so the innate immunity, which is also known as non-specific or non-adaptive immunity, is the first line of defense against infectious organisms and do not depend on previous exposure. That is, the immunity existed before encountering the microbes. And these non-specific host responses provide an effective barrier that prevent the microbes from penetrating, inhibit, or destroy the invader if it gains access to the tissues, as well as eliminates or neutralizes any toxic substances elaborated by the infectious agents. Okay, so as I told you, some of the barriers such as phagocytic cells, neutrophils, macrophages, NK cells, physical barriers also. Okay, chemical barriers also, physical barriers such as skin, and chemicals such as TS. Blood proteins, some of them are such as complement or other mediators of inflammation, and the cellular factors such as cytokines. So now we are moving forward with the acquired or adaptive immunity. Okay, also known as specific immune response. The more effective will be its different agents against the various other microorganisms. And this acquired response also retains the memory of previous exposure to the antigen, so that the next time it encounters the same antigen, the more stronger will be the immune response. The acquired immune response takes several days to develop, that it develops rather slowly, but once it develops, the immune system is incredibly effective. Okay, so basically this adaptive immunity is a defense system that protects the body against the pathogenic microbes and other types of diseases. Okay, and this type of immunity is divided into active and passive immunity, in which the passive immunity is basically divided into further two types: passive naturally acquired, passive artificially acquired. For one of the examples of naturally acquired passive immunity is the maternal immunity, and the passive artificially acquired is basically one of the example is the antibody transfer. In the case of active immunity, natural acquired, okay, and another one is the artificially acquired through the immunization process. Okay, so what is the basic difference between active and passive immunity? Okay, so in the case of the active immunity, okay, produced actively by host immune system, whereas passive immunity, this is basically it is received through passively where no active host participation. In the case of active immunity, it may be induced by infection or by immunogens, whereas in the case of passive immunity, ready-made antibodies are used. Okay, and active immunity is effective only after a lag period, but passive immunity, okay, it provides an immediate immunity. And in the case of active immunity, immunological memory is present. In the case of passive immunity, no memory will be there in the system. Okay, so now comes the concept of herd immunity. What do you mean by the herd immunity? So basically, herd immunity is defined as the overall immunity of a community or a herd towards a pathogen. And herd immunity plays a vital role in preventing epidemic diseases. If the herd immunity towards is good, that means the large population of the community are immune towards a pathogen. Hence, the epidemics are less likely to occur and eradication of the disease may be possible. There are various other factors which contribute to create herd immunity. For the some of the examples are immunization program, type of population. Okay, also it also can be enhanced with the more study regarding the herd immunity. Okay, now comes the basic difference between innate and adaptive immunity. In the case of innate immunity, which is a quicker one, where on the onset of the infection, okay, body recognizes the pathogens by the senses, activation of cells and inflammation, and finally removal of infectious agents. Whereas in the case of adaptive immunity, a long term, which is a specific one, okay, on the onset of infection, it stimulates B cells and T cells in the lymphoid organs, and expansion of training of affected T and B cells, migration to the infection site, and finally the removal of infectious agents. Okay, now I want to ask Sagar sir to continue the lecture.
Okay, so we are going for the structures, functions, and the properties of immune cells. As sir discussed about the immune response, so there are some immune cells which are directly affecting to our immunity. So first of all, we will go for the stem cell differentiation from the bone marrow. Okay, so in bone marrow, we will find out the stem cells, which is hematopoietic, and which will be divided into two types, which is a myeloid and another one is lymph node. So there is also a subclassification which is undergoes as a megakaryotype, eosinophil, basophil, erythrocytes, monocytes, and neutrophils. As we all know, these all kind of the cells are having some different properties and different functionality for the immune system. And when we are talking about the lymph nodes progenitor cell, there is a T cell, B cell, and natural killer cell. And B cell also produce a plasma cells. While we are talking about these eosinophils, basophils, erythrocytes, monocyte, and neutrophils, there is also the platelets and the dendritic cell and macrophages, which having a different characteristics, which are going discussed here. So when we are talking about the basophiles, firstly, so what is a basophile? Basophile is a particular type of phagocytic immune cell which has the granules, which has their granules, which will help us to get a interactions between a releasing of the histamines during allergic reactions. Now we are going for the lymphocytes. What is the lymphocytes? The lymphocyte is a one type of the cell of white blood cell, and which develops a bone marrows and also form a antibodies. So now we are going for the dendritic cells. Dendritic cells are the APCs. What is APC? Antigen presenting cells. Antigen presenting cell. And this antigen is combined with major histocompatibility complex, and it will also present to activation of T cell and B cells. Now we are going for the eosinophils. So eosinophils is a type of the immune cell which is also known as a leukocyte, and they will help to fight against infections of the cell. And now we are going for the T cell. T cell is a type of the white blood cell, and which is having a capability to fight against the infections. And now we are going for the natural killer cell, NK cells. So what are the NK cells? They are the first responder of our immune system, and they will first response, and they will form a activation of our immune system. So in the next slide, you can see they have the structures of the stem cells, T cell, B cell, natural killer cell, and dendritic cells hereby. And you also get to know in the next slide, it's a neutrophil, eosinophil, basophil, mast cells, which is having a capability to inclusion into the degranulating, and also the macrophages. They all are having a differential proportion in our immune system, and all are the essential for our immune system. And now I am giving Patni sir, asking to Patni sir to continue the structure, function, and property of immune organs. Thank you, sir.
Thank you, Sagar sir. I'm going to continue with the structure and some of the functions of the and the properties of the immune organs. I'm just going to cover only the basics of these immune organs. Okay, so basically lymphoid organs. Okay, basically lymphoid organs. In this diagram you can see the immune system or the lymphatic system of our human body. You can clearly see the lymphatic system of our body, which I have indicated is the thymus, Peyer's patches, lymph nodes, spleen, and bone marrow. Okay. Now moving forward with the lymphoid organs. Lymphoid organs are basically divided into two ones: primary lymphoid organs and secondary lymphoid organs. Okay, and which are the primary lymphoid organs? Thymus and bone marrow. The diagram which can be seen in the screen here. Okay, what is thymus and what is bone marrow? Bone marrow, basically, it's an progenitor of T cell and B cell, basically originate in the bone marrow, and the thymus is developed in the embryonic life from third or fourth pharyngeal pouch, and it is highly active at birth and reaches peak size at puberty, and then it degenerates. And basically, structure of thymus has basically two lobes. Each is differentiated into outer cortex and an inner medulla. And cortex basically is densely populated and contains numerous thrombocytes, epithelial cells, okay, and nerve cells. And next comes medulla, which is sparsely populated and which contains few of the thymocytes, epithelial cells, dendritic cells, and Hassel's corpuscles. And there are also thymic hormones such as thymulin, thymopoietin, thymosin, which are produced and aid in the development of the T cells. Now comes the secondary lymphoid organs: spleen, lymph nodes, MALT, and GALT. Okay, what is lymph node? Lymph node are small bean-shaped organs which is divided again into cortex, medulla, and paracortex. The cortex contains lymphoid follicles which are mainly of two types: primary lymphoid follicles. They are present before the antigenic stimulus. They are smaller and contains resting B cells. Next comes the secondary lymphoid follicles, which are formed following the contact with an antigen and contains activated B cells, for example, plasma cells and memory B cells. And they are divided into central area and peripheral zone. Next comes the spleen, is basically which is a second, it's the largest, sorry, it's the largest secondary lymphoid organ divided into two compartments: central white pulp and outer red pulp. White pulp has two parts: periarteriolar lymphoid sheath, which is also known as T cell area, and the marginal zone, which is known as B cell area. In the case, red pulp contains sinusoids which are filled with RBCs, and other and older and defective RBCs are basically destroyed here. Okay. Next comes the MALT. MALT are the basically lining, okay, which is also known as mucosa associated lymphoid tissue. Okay, and it's a lining present. It's a present lining the mucosa of the intestine, respiratory, and urinogenital tract. MALT in the intestine are the best studied MALT present in different layers of the intestinal wall also. Okay, so now next comes the GALT. Okay, GALT basically it's also known as gut associated lymphoid tissue. It is known as gut associated lymphoid tissue. Okay, submucosa contains Peyer's patches, which is composed of lymphoid follicles, lamina propria, which lose clusters of lymphocytes, as well as the macrophages. And both the layers MALT and GALT also contains epithelial layer, which contains few specialized lymphocytes, M cells, okay, and as well as they are lined by secretory IgA, which provide local or mucosal immunity. Okay. Now, Sagar sir, can you please continue?
Yes, sir, please. Just do it. So, in the last, what is the characters of immune system? So, what we studied, what is the immune system has a characteristics. So, mainly there are the three characters which are essential and important for the immune system, which are given here, which is a self recognition. So, what is a self recognition? Recognition between antigen and antibody यानी कि or recognition of non-self. Foreign particle कहो है. What is a foreign particle or the foreign cell which is inserted in our the body? It is a considered as a self-recognition. Specificity. Specific antigen antibody interactions are occurred for the immune system for the more healthy immune response. And the last one is a memory. So when the we are adapted through the vaccination or somehow through the immunization process, we will get to know the memory. These 3 points: self-recognition, specificity, and the memory are the known as for the characters of the immune system. And that's all for today. We will post more videos on another topic too and another chapters too on upcoming week. So stay tuned with us, me and Mr. Patni sir. Okay. Don't forget to like, share, and comment down which portion you would like to learn in the next video. We would like to share this. Thank you, everyone. Thanks for watching.
Thank you, Sagar sir. Please like, share, and subscribe my channel as well as Mr. Sagar Sonara's channel. Thank you, everyone.